spills up at Kamehameha Hotel. There was another sewage spill by Huggo's Restaurant. There was a sewage spill at Keauhou Bay. And the state comes out and takes samples and they say, oh, it's a little high, but it'll be okay. And recently they've had to put up signs. This is new. Why did they have to put up signs? Because it's the law. It took the Surfrider Foundation in Kauai, Dr. Carl Berg, friend of mine, to get the EPA involved, to get the state to do its job. You're going to find out really quick, the State Department of Health is not high on my fans list. They need our help. They see the world through very narrow blinders. So let's talk about testing the water. We've got a problem on Hawaii Island. Water flows downhill. And I have letters in my files where the state has said, no, the groundwater doesn't go into the ocean. When I was a tourist here, I hit this cold water that I couldn't see through well when I was snorkeling. And I thought, what's going on? And then we learned. We have no rivers on this side of the island. Hilo has 212 rivers and streams. We have none. And yet our mountains get 80 inches of rain, 100 inches of rain. Where's it go? According to the state, there's a dam at the shoreline. But we know that water flows downhill and water flows to the sea. Dr. Greg Glenn, a great geographer at UH Hilo, has studied our coast, and he has found that for every mile of our coastline, 1 to 3 million gallons of fresh water flow into the ocean every day. The fishermen know where those upwellings are, because it's oxygen-rich water, and the fish like that. The Hawaiians knew where they were. They would take gourds down and fill them underwater and bring them to the surface. It was all fresh water. Our Ancolin pools, the Queen's Baths, are all fresh water flowing to the sea. Then we decide, because public law says we should, we need to put our toilet water in the ground. And here's a picture taken at Puako by the residents up there where they put fluorescein dye in the toilet, flushed it, and within hours the ocean turned green. They have a real problem up there. A lot of people living about 8 feet above sea level. And everybody's got some type of cesspool or cesspit. Well, there's blame to go around. There's 50,000 of those things on this island. And exactly where they are and what they're doing, nobody really knows. But the state's happy because it's out of sight and hence out of mind. And maybe it's safer, we don't really know. But we have a problem that needs to be fixed because it ends up here, sooner or later. So in aggregate, on the whole Kona side, we're looking at about 30 million gallons of wastewater that's being discharged into the ground every day. And the fate of that water is largely scientifically unknown. There are people who will tell you the ground filters it. The nitrogen is gone. I've read an EIS on the proposed condominiums down here, Keauhou.
Timeshares.
Yeah, and the expert says, well, that the ground binds the nitrogen and phosphorus, and none of it gets to the sea. And at a meeting, I asked him, I said, can you cite the scientific references for that? He goes, oh, we got it somewhere. Yeah, in his head. And so we're going to talk about microbes today because that's one of my favorites. I've been working in environmental watershed microbiology for over 30 years. I got my training on Tomales Bay, California, a small residence called Point Reyes Station, and maybe 10 or 15,000 dairy cows in the watershed. Water flows down there, downhill there as well. And I didn't enamor myself with the dairy farmers, but that's okay. We know virtually nothing about the fate of these microbes in the environment. Attenuation is the word we use. To attenuate a microbe means to eliminate its capacity to make disease. Now, here's a myth I want to kill. You read state literature, you read EPA literature, textbooks. Human feces is a disease epidemic waiting to happen, right? There's cholera, there's hepatitis A, hepatitis B, hepatitis C, there's salmonella. Yes, those things occur from time to time. But if I were to take a sample from all of you today, I would probably find none of those because you're healthy. It's the sick people who shed the disease organisms. You would not want to drink the surface water today in Yemen. Why? There's a cholera outbreak. People have cholera, excrete millions of the bacterium Vibrio cholera. But when you're healthy, you don't. And so we have caused a lot of fear about human waste and disease. 99.99% of the trillion bacteria in your colon are harmless. Harmless. And in fact, they're actually beneficial. And that's a whole nother story that I'm involved in, but we don't talk about that today. We don't have time. Okay, there's nitrogen and phosphorus in human waste. The nitrogen is in the urine. There's very little nitrogen in feces. There's phosphorus in the feces and a tiny bit in the urine. Nitrogen and phosphorus are fertilizers. I have a confession. I pee on my lawn a lot. So as not to put it in my cesspit. Because the grass like it and the grass will grab it. Fortunately, I have no neighbors. When I move to town, I may have to change that. Now, the state is advocating, in some cases mandating, we switch from cesspits to septic systems. I have jumped into the science of septic systems. There are people that say that Rick knows his, and unfortunately I do. And what the science says, the contemporary science, is septic systems offer a smidgen improvement. A smidgen. And in my view, it is inefficient and cost ineffective to require people in Hawaii to put in septic systems. The state claims magic in their website. I have challenged that officially to the director of the public health department and the governor, and they refuse to answer my letters. Water drains to the sea. We've all established that. We don't have to argue that, although the Department of Health will. We use monitoring tools that are antiquated and misinterpreted by intent. The monitoring program we have for recreation water is poorly funded. They get $900,000 a year from the EPA under the Beaches Act legislation to do the program they have, and it's not enough. And it's poorly staffed. And I'll have something to say. And most importantly, the risk associated with the numbers they generate is completely inappropriately interpreted. In other words, we don't know the risk, one way or the other. And I get a lot of phone calls from parents of children. Is the water safe? And I have to say, you know, I don't know. And this is my business. We should know. And I'm embarrassed to say, I don't know. So, we could talk about the history of fecal indicator bacteria a long time, but I'm not going to. We don't have that time. But it's interesting to me, the acronym fecal indicator bacteria is FIB. And so these bureaucrats are not really good at public relations, you can tell. But the implication here is that we can measure a surrogate. We can measure something in the water that we think is associated with human feces because it's easy to measure, and under some situations there seems to be a correlation with disease risk. And I'll explain how that happens. But the organism they're using today, as stipulated by EPA regulations. Now, a footnote on that, I was speaking with the EPA last week, and they said, Dr. Bennett, we know you're concerned, they know me, and they said, we admit that of all the tests that we do in the EPA, enterococci is our least favorite because it is so imprecise and has such a poor correlation with risk. All EPA regulations are based upon risk assessments. And to do an accurate risk assessment, you have to be able to measure precisely the thing that's causing the risk. They're really good at doing mercury and PCBs. They're terrible at enterococci because of the nature of the test. They don't have the funding to develop a new one because you know what's happening at the EPA level. It's been pretty much killed. But we're stuck with it. This is what the state is stuck with. It first got its use in 1940. They called it fecal strips at that time. They're not fecal strips. In 1970, EPA did this risk assessment thing on coastal and lake waters where they knew there was a sewage outfall. We don't have any sewage outfalls in Kona. Hilo has one. Honolulu has a big one. We don't have one. So that's deficiency number one. Okay? Deficiency number two is that the enterococci is a genus. And in that genus there are 50 species. And so this test detects species of enterococci that are not human oriented. And that's a huge problem. It's called false positive. Okay? Dr. Fujioka in Honolulu UH discovered that enterococci grew in the streams in Manoa before it got to the campus. And then after it got to the campus, a little more time it got to the sea, there was a lot. And he concluded that this thing grows in the streams and was really the first one in Hawaii to raise concerns. Deficiencies in this fib. There's 50 species of enterococci, only 2 of which are low-level pathogens for humans. They don't cause much disease in healthy people. They grow in clusters and in chains. And so if you're going out here and taking a water sample and you shake it up and you put it into your test system and you're trying to count individual bacteria and there's clusters or clumps, that clump gets counted as 1, when in fact it may be 10. That's a huge error in this estimate. So they're hard to count. They persist in the environment. Well, that's clever. Hope it doesn't fall. And it does not correlate well with the presence of virus in the water. If you get sick today from recreating in our waters, it won't be a bacteria that made you sick, it will be a virus. Have you heard of this one called the norovirus, also known as the cruise ship virus? It goes through a cruise ship like manure through a duck, and it freaks them out. So we have moved the disease risk from drinking water, this is probably not in Kona, but in Hawaii we built this thing called the Sand Island Treatment Plant that processes 90 million gallons per day of sewage. And then several years ago, they were putting advanced primary sewage in the ocean. And the EPA had to stop them from doing that. And I challenged then Mayor Hanneman on this, and he said, well, talk to my environmental people. They'll tell you it's okay. So I talked to the environmental people, and they said, we're not sure what the mayor's talking about. So I got the political two-step on that one. So the test that we use today, when the state comes out, is called the Ginter Alert. And it is a EPA-approved test for freshwater and marine waters for recreation. So you take a sample from the water, you shake it up good, and you pour it into this tray that's got 51 wells in it, and then it goes through a sealer. So now each of those wells is sealed off from one another. Incubate it 24 hours, put it in under a blue light, and you count the wells that are glowing. You take that count and then apply it to a a table and it gives you an estimate. Okay, here's a picture of enterococci growing in media and then put under a microscope. There's very few, if any, that are single colonies. They're clusters. They're chains. And so the nature of this microorganism doesn't lend itself well at all to use the most probable number counting method. So let's talk about that. So they take a sample out here, they take it back to the lab, they process it. In fact, the lab is here in Plymouth. There's a contractor, Monica Duncy, and the state brings her the samples to her commercial lab, and she runs these. She lives up Palisades area. And you count those cells, and let's say here we've got 5 glowing cells. The most probable number is 5.3, but the 95% confidence interval, that is to say 95% of the time, so you have a new job.
Yes.
Put something on the back leg. Can we hook it at the top? Put something heavy on the back leg that sticks out.
That might help. Oh, this is a big sail right here.
Yeah, it's a big sail.
Okay, so the number really, well, let's use my illustration here. Let's say we've got a 17.8, which is right here. The 95% confidence interval is a low of 10.8 to a high of 29.4. And so what we know is there's a range of microbial densities out there in the water, and we're 95% confident about that range. The state, however, reports this number as an absolute. So anytime you see a number of enterococci in the media, you have to know the range is much bigger, and it's somewhere in there. And this is, counting individual bacteria is not easy. And this MPN method was developed, 80 years ago or more, and it's not very good. But it's the law of the land. The U.S. Beaches Act is being implemented here in Hawaii because of what my friends on Kauai did with EPA. And here is the number that we're worried about. When the count, the most probable number count, hits 35, that is reason for concern. Regulatory concern, not public health concern. And if in a period of 30 days, the collected aggregated data averages at 35, then they're required to post a sign. If in that 30-day period 10% of the samples exceed 130 on the MPN, they have to sign immediately in a little more serious kind of sign. But they're just signs. They're not beach closers, they're just signs. Okay? And this estimate of gastrointestinal illness, 36 per 1,000 recreators, is based upon recreation water where there is a sewage or storm drain overflow. And that's where they are able to do this risk assessment thing. We don't have those here. And we don't know which cesspool or which cesspit is draining into the coast where. We just don't have that technology or that ability yet. But this is the law. And so we're going to see more signs here. But don't freak out. Because this probably seriously, greatly overestimates the risk from bacteria and completely underestimates the risk from virus. But still don't freak out. Why? Because we're not seeing clusters of disease. If 10 people in your canoe club had nausea, vomiting, and diarrhea after spending some time on the water, that'd be a disease cluster. And that should be reason to call the health department and say something's up. And they'll go, well, what'd you have for breakfast? It's probably going to be that. Well, we didn't have breakfast that day. So here's a little number game they play. This is a simulation I did on my computer representing the MPN numbers for what could be a little sewage spill, like the one that occurred here after the tsunami when that pipe broke. Okay? So for a few observations, you got some pretty significant numbers, and then subsequent days, you got a bunch of less than ones. And this MPM table will give you a less than one. Anybody familiar with Excel spreadsheets? How do you average less than one? You can't. You put a 0 in there and you get a divide by 0 error. You can't. So what the state of Hawaii does said, well, we can't summarize those data, so we'll just call it 2.3. Arbitrarily. Out of thin air. And I've consulted statisticians and they've said, no, you can't do that. You have to use what's called a non-parametric assay. I'm sure DOH has never heard of it. And so what I did was simply throw them out because it's below the detection limit. And when I take the mean of these, I get 48.9. When I take the mean of these, I get 23.5. One is violative, one is not. That's what I call the mathematical two-step. Now the state and the EPA both take what's called the geometric mean, because you've got some really high numbers and some really low numbers, and statistically that's appropriate. But again, when you include the 2.3s or throw them out, the geometric mean is 46 violative or 2.3 non-violative. Figures don't lie, the liars figure. Am I blaming them? Yeah, I am. Okay, ocean users are the proverbial canary in the coal mine. All you canaries, nice to have your attention today. Research in Oregon. Anybody ever surfed the Oregon coast or been up there? Not a lot of people. Not friendly water to play in. But people do. And what they found is that on average the surfers surfed 77 days a year. These are brave souls in 5 mil wetsuits, huh? And they consumed 170 mils of water per surfing day. That's a lot. But if you've wiped out on a wave that's bigger than 2 feet, water gets forced into every orifice under great velocity, and you end up swallowing a fair bit, obviously. And so what they saw in Oregon was only an almost indetectable increase in illness. And some of that illness could be from swallowing too much salt water. The salt water will make you vomit. And so that's not too conclusive, other than they swallow a lot of water. But what caught my attention here lately, because I personally have experienced ocean skin infections, is that ocean users are 5 times more likely to have a skin problem, a rash, a pimple, an infection, than non-users. Now, admittedly, this is in Florida water. And if you've been in Florida beaches in the last decade, there's a whole lot of people living there. And all their septic systems and sewer systems are on coral sand. Flush, boom. And so is this risk appropriate for Hawaii? My guess it is, because we don't have soil, we have fractured lava, we have some coral sand, and it ends up in the water. So when a kid like this guy here, this is taken in the winter, the shore break at Napo'opo, it gets real dramatic, and these kids just go out there and get beat up. I'm sure he hit the sand really hard. And so he's at some risk. Now, I am getting reports from various surfing organizations up and down the coast that people are getting rashes. In particular, children, non-surfers, who are playing in the sand are getting rashes. I don't know what it means yet, but we're going to pay attention to that. So, sand. 20 years ago, I was sampling water in a stream that flowed into Tamales Bay where they grew oysters, and I had my technician take our pole and go down to the bottom of the stream and get me some mud. We measured the bacteria in the water flowing by quickly, and we measured the bacteria in the mud. The mud had 5 times, 5-fold greater. And I thought, that's weird, maybe it was a mistake. We did it again, same story. Well, we now know, 20 years later, that the sand, this little girl is the baby of a friend of mine playing at Manini Beach. We now know that the sand offers a place for bacteria to live. Bacteria don't like to live in the water. They like to fall down and attach to things. And the bacteria get in the sand where they're out of the light. So researchers at Manoa just a couple of years ago did a laboratory experiment looking at survival of bacteria in the water column above the sand, and then survival of the bacteria in the sand. And here's what they found, is that in the water the bacterial numbers dropped precipitously in about 6 days. In the sand, they stayed fairly constant for over 3 weeks. Down in the sand, out of the sunlight, and probably in a place of more nutrition for the microbes. Are they growing? We're not sure. Are they surviving? Absolutely. And so this is where the sun comes into the story. This is where Dennis's hard work and research has helped us understand and raise our concern. This is my paddle. This is one of your boats. Karen Kemp said, "Come down, Rick. We want to show you the water." So I jumped in with my paddle. I stuck it in the water, fairly close to the shore, but not so close we're going to hit something with the canoe. I can't read the name on my paddle. Can anybody? I've had so many paddles I can't remember which one it might be. And we were concerned because this is not typical. This is not normal. This is not healthy for a bay or inlet like this one. And here's what's happening. We're getting excessive nitrogen and excessive phosphorus equaling algal growth, equaling phytoplankton growth, causing a increase in turbidity, as Dennis documented so well. One of the reasons our water is generally very safe is because of our friend the sun. That same energy that gives me skin cancer time and time again, because I'm Norwegian by ancestry, where the sun doesn't shine a whole lot, the UVA and UVB rays penetrate the water, hit the DNA of bacteria, and break it. It's tantamount to tearing up the blueprints on the home you're trying to build. When the DNA is disrupted, the bacteria die. Not only that, low dose ultraviolet will injure bacteria. They're viable but not culturable. So they'll grow, but they won't grow in the Alert test. So they're alive, but they won't grow. Higher doses will kill them. And as the light transmission drops off, the ultraviolet drops off dramatically. So in the surface 50 centimeters of water is where you get good penetration in clear water. When water gets turbid, you're not getting good sunlight penetration. Make sense? It's all connected. This is like living ecology here. Now here's a young man named Neil Mukai. He's got a 2-year degree out of Hilo, and he was hired by the DOH to water sample. You may have seen Neil out here, knee deep in water, which is protocol, and he takes a sterile vial. How do I know it's sterile? It's got autoclave tape here, that when it's autoclaved, when it's cooked in the laboratory machine to sterilize, the tape gets stripes on it. And then he dips his bottle in the surface of water, and you're not supposed to allow the water to hit your fingers because your fingers are covered with bacteria. So he's very carefully getting water from the surface, the very surface of the water, where the sunlight is the strongest. And he's doing it all day long. So in Miami Beach, here's the research done. They looked at bacteria counts collected in surface waters between 9 a.m. and 2 p.m. And they found consistently, over a period of several months, that there was as much as 100, minus 1 is 100-fold decrease in the indicator bacteria counts from sunlight. If we want a better indication of what's going on in our water using indicators, we should be sampling before 9 a.m. and after 2 p.m. Now what does that do to this man's work day? Cuts it in half, which means he has to work twice as many days to get the samples. This is not going to fly with the administration. But to be scientifically valid, that's what needs to happen, or find a better test, which is where we're going. Okay, let's change gears and talk about another organism that's dear to my heart because I almost lost a finger to it, Staphylococcus aureus. How many of you have had a staph infection? Okay, a number of you. The summer that I almost lost my finger, 10 of my paddling buddies at Keauhou had staph infections. One man, and you might know Tim, had to be hospitalized twice for IV therapy. He came within a gnat's whisker of losing his leg. And we think that staph was flowing into the little bay where we launch our canoes, on that little slippery boat ramp like yours. There's a preschool right there with a septic system. And it flows directly into that little bay that has very poor circulation. And so that's my guess of where it came from, but we'll never know. For some strange reason, and maybe it's this, Hawaii leads the nation in staph infections. And state public health people have thought, well, what is it? Their diet, their genetics. They failed to realize that a lot of us in Hawaii spend a fair bit of time in the ocean. It's part of our culture. And to date, they haven't looked at it. In fact, Staph aureus infections that a physician sees are not reportable to the state. If you have Hansen's disease or tuberculosis, it's reportable. But the clinicians that I'm speaking with are seeing at least 1 or 2 cases of staph skin infections a day in their clinic. A day. And that should be alarming. So what the research has found is that it's in the sand, more so than in the water, and Staph aureus, a count of 40 in a square centimeter of skin surface area, represents a 20% infection risk. It has a very low correlation with enterococci in the sand. That is to say, when the enterococci is high, the staph can be low. When the opposite is true, can also happen. Correlation is poor. So it's moving independent. Where does the staph come from?
People.
Okay. 30 to 40% of us carry it in our nose, and about the same percentage carries it at the other end. And so when you shower, it goes into the wastewater. When you recreate, it goes into the ocean. It likes salt. It uses salt for its competitive advantage with the other bazillion bacteria that normally occur in the ocean. So a little science to show this. This is some California beaches, including Avalon Beach and Doheny Beach. Here is the counts of Staph aureus in the water. Here's the counts in the sand. And this is a log number of about 5. So it's about 150 is the count. And 5 is up here. So the sand's got more staph in it. So when you play in the sand, when the kitties play in the sand, they are statistically likely to encounter more staph. Does that mean they're going to get infected? Not necessarily. If they have a wound or a pimple or a scratch, the risk goes up. So we're getting to the end here. Here's some recommendations. Anybody in healthcare here? Nurse, doc? Universal precautions. What is the assumption you have to make about all people?
Everybody's infected.
That they're all infected. When I first learned it, I go, no way. So if you're a first responder and you go to the scene of an accident and there's blood everywhere, you have to assume that blood's contaminated. So what do? You put on gloves, and if they're vomiting stuff, you put on face shield and goggles. Because like the lady said, you have to assume that all people are infected. I'm proposing, and it breaks my heart to say this, that we have to assume that our oceans are always contaminated and behave with a little bit of caution, but not fear. Okay? Some cautionary items. Don't recreate in stormwater. How many times has Kamehameha 3 flooded, come ripping down the driveway, jumped the curb, and into the bay? That's stormwater. Stormwater is only a smidgen better than raw sewage. Sewers leak, storm drains flood. In San Francisco, when they got a big storm, the sewers would lift the manhole covers, those big heavy iron covers, and raw sewage would run down towards the Embarcadero. Avoid floating scum. I learned this the hard way. I'm a swimmer, and I was swimming laps in Honolulu Bay. And over in the northeast corner, which was the wall I would stop at and turn around and go back to the island, I looked up, there was all this stuff floating in the water. It was brown, it was particulate. And I took my hand, and it was almost like greasy. And then I realized that during low tide, the cesspool on that house, that right there in the corner, would drain into the ocean. When they do laundry, there's big standing bubbles on the ocean from the detergent. Don't swim in scum. Open wounds do not heal in salt water. My mother used to say, go in the ocean, Ricky. It'll help heal your cuts. It's not true. That strong salt water kills tissue. It'll get white. You've seen that on a wound where it gets white? Okay? That white tissue is bacteria food. It is devitalized tissue. It's lost the ability to resist infection. And I saw that happen on my finger. It got all white. And then when the blisters started occurring on the back of my finger, I said, oh, I gotta go get some antibiotics. Vigorously clean and medicate wounds created in the ocean. How many times? You know, you're paddling hard, wow. And the knuckles on the gunnel, right? Yeah. Don't ignore those now. Wash them, soap and water. Don't use disinfectants. Don't use iodine. Don't use hydrogen peroxide. It kills tissue. Kills tissue, makes bacteria food. What you do in hospital today, you get a wound, they've got this pressure syringe. The doctor or nurse puts on a face shield, and they take this pressure syringe and they blast your wound. It hurts. You don't have to blast your wound, but take the kitchen, all of our faucets are, on that little cord thing. Turn it on full blast and just irrigate. And then put some Neosporin on it or something like that. But irrigation is the key. Do not ignore pus-filled little pimples. A number of years ago, during the Liliuokalani race, a number of people came from the mainland to race. And I was talking to this man and his wife. I said, if you get little pimples on your legs, don't ignore them here. And a couple of days later, he said, we've got little pimples. And let me see. And it's like a little mosquito bite with a little dome that's pus-filled. And he went to the clinic, and the doc said, that's probably staph. And started him on antibiotics. It was just total irony that happened. So don't ignore those pus-filled pimples. If it gets larger, seek medical attention. Wash off. You've got a shower here? I'd use it. Especially the kids. Use it. Just rinse off. And then in terms of gastrointestinal disease, if it lasts for more than a day or two, the norovirus is called the 24-hour flu, because usually in 24 hours you stop vomiting, you stop having diarrhea. But it persists. That's a bad sign. And you really should see medical attention. And then the most serious one is you get a little wound, and around that wound you start getting really nasty bruises. There is a bacteria in the ocean. This is an ocean bacteria, not a sewage bacteria. It's called Vibrio vulnificus. And people who are immune compromised, people who have high blood iron levels are really at risk. And this will kill in 24 hours. There was a guy who fell into the Aloi, and this organism got into his body, and he was dead in 24 hours. And so again, swelling of a limb, with or without severe bruising, get there ASAP. Okay, enough scary stuff. Let's talk about the future. We now have the scientific ability to speciate the microbes in our environment, in our gut. Did you know that there are 1 trillion bacteria in your colon? 1 trillion. And there are 1,000 species in there too. 99.9% of which are harmless. They are doing very important things. If you haven't heard of the Human Microbiome Project, look it up on the internet. Those organisms are as important as our liver to our health. And we just figured this out in the last 10 years. Well, using that technology, we can take a water sample and send it through this DNA analysis like they do at UC Berkeley. And you get data that is a mountain of data, but there are software programs that'll make sense of it, and this is one of those. So here we have sewage, here we have a pig. The dots in green represent individual species of bacteria. And so there are literally thousands here in sewage that are green. When it's green, that means it is unique only to sewage. Okay? The commonality between pig manure and human manure is expressed here in the blue dots. The commonality between all of these species is expressed here in the pink dots. And so today, we can take a water sample out of the bay, run it through this process called the phylochip, and it will tell us this. When we get a human signature, that is incontrovertible evidence that there's human fecal bacteria in the water. Now, does that mean there's risk? Some. Again, most of these microorganisms are not pathogenic. But this will end the controversy that we hear in the media here. Well, you know, we're finding enteric oxide, but it's probably from cows or dogs. They say that all the time. Now we can tell. So here's some interesting data. I'm almost done. This is done by the group out of Berkeley. Dr. Gary Anderson here is the man that we are talking with about doing this work here in Kona. And on the Russian River of California, north of San Francisco, there is a beautiful river that people play in all summer. I've done float trips, I've done party trips. It's a great place to play. So they came in there in the summertime, couldn't find any human signature in the river. None. Wintertime, different story. All those homes are on septic systems as required by Sonoma County Public Health Department, and they found a fecal signal in samples of the river where the indicator counts were below the EPA precautionary standards. That is to say, a signature of human fecal bacteria was present when the indicators were not. Okay? I'm excited about bringing this here because for once and for all, we'll be able to say, yes, there is evidence of a human signature in our waters, or there is not. I suspect we're going to see it. And the question is where and how much, and we can answer that question. So we're proposing a study that's going to look at 10 beaches over 9 months, 3 separate samples, and we're going to have mostly urban beaches, and we're going to have one shoreline down south, above Mililii, where no people live. The only thing there is goats. Too many goats. So I'm excited about this. And so our action agenda, and Dennis and I are working, some other members of this canoe club are going to be participating. We want to raise the money, about $20,000, to do this project with Dr. Anderson. And so that stands on its own. The other thing we want to do is identify, which I've already done, and close cesspools that serve public beach restrooms. The biggest offender of all is Hapuna Beach. They have seepage pits for those toilets. And a lot of people use those facilities, and a lot of people play in those waters, and those waters are frequently showing signs of pollution from a variety of sources. At home, one of the easiest things you can do to decrease the flow from your home to the sea is don't put your gray water in the septic tank. Don't put your gray water in the cesspit. When I built my house, I cheated. I have two waste systems. The toilets go into the cesspit, everything else goes into a tank, and it irrigates my avocado orchard. So the only water that's driving down towards the sea is 2 or 3 gallons a day of toilet water. And then at the city level, we are moving, I say we because I'm on the Environmental Management Commission. In fact, they mentioned my name today in the paper about some noise I'm making. The wastewater at the Keauhou treatment plant, the intent is to reuse it through gray water irrigation. The intent, a stated intent. I'm going to hold their feet to the fire. And the mayor and I are on first name basis, and he has told me, he says, if you see something you don't like, you tell me. So I guarantee I will. What the state and the county can do is to do some more development work on enhanced, advanced treatment units. This is a little waste treatment plant that you can put at your house that takes the nitrogen out of your wastewater, sends it off as a gas to the environment. 80% of what we're breathing right now is nitrogen, so it's not a greenhouse gas. And the wastewater is disinfected with ultraviolet light and then reused around your home. A little bit pricey right now, but I think once this gets commercialized, it will effectively replace, and so we don't need to put in new septic systems. And then the last thing is, let's continue to build a citizens monitoring program. I was aware of a citizens monitoring program at a beach in Santa Cruz called Steamers. It's a famous surfing place. The warning signs were up perennially and basically rusted to the point you couldn't read them. But the Surfrider Foundation there did their own microbiology several times a week, and it got published in the newspaper. So after a storm event, the counts would go way, way up. Surfers didn't stay out of the water, and eventually it put enough public pressure that they had, the county of Santa Cruz had to build a new waste treatment plant for the town of Santa Cruz. Had it not been for that citizens project, it probably wouldn't have happened. Okay, so let's make them proud. These are my mo'opuna. They were visiting here a few years ago, my grandkids. My daughter's a practitioner. She goes, Dad, is the water okay? And I said, I think so. She goes, it better. And so every time that I talk about our water quality, I think of Aiden and Macy, because I'm standing for them and all the children of the future. So thank you for your attention in this hot. What we can do is to go out there and take some samples and look for things like ammonia. Ammonia is easy to test for. And if there's a lot of ammonia in the ocean, that means something's leaking. And Dennis, we can get an ammonia test kit.
Well, and there, there's little bubbles and scum on the water.
Yeah. Okay, so something's going on. I've asked to tour that plant. I've invited Dennis to join me, and I'm going to have William Chukarski, who's the director of the environmental management unit in Hilo, and I trust Bill, and he's a square shooter, very different person than the previous director, who's gone now, thank goodness. But I hear your concerns. I got a phone call from Pat Cunningham the other day. He said it's stinking out here, and so yeah, I think ammonia would be the easiest way to say there's something wrong, and with some positive ammonia results, we can go to the state and say, go find out where this ammonia is coming from.
Is that something you would do outside of this proposal you've made, or is it something?
Well, it's something we could do right now. If you suspect waste, fish waste, human waste, garbage is being discharged into the sea, it will decay. It is decaying, and it will release ammonia. Okay? Why am I finding? No, I'm not finding it. NELHA reports high ammonia levels offshore from their facility. What's decaying there? Turns out the waste from the shrimp farms and the waste from the abalone farms is being put into what's called seawater return trenches. They were intended for warmed seawater when that NELHA thing was about doing OTEC conversions. And so they're discharging wastes into the ocean through those trenches, and it's showing up as ammonia. And the state's not doing anything about it. So yes, we can generate some evidence that the state couldn't ignore. Because you don't find ammonias in our water.
So Rick, do you suggest we take a little jars with us when we go past and use it?
Well, once we get an ammonia kit here, because you need to do it quickly, because ammonia quickly is converted by bacteria in the water to nitrate. And nitrate is hard to measure in seawater for a variety of reasons. But ammonia is easy. And so if you have the kit, get some little snap vials. You can get them at Payless. And they just open and they snap closed really well, and then you can bring them here and then run your ammonias. So that's an option. I can work with you guys and help you get set up.
Last Saturday there was oil on the water, really thick, black, gunky oil, just out beyond the Sheraton.
Okay, grab a sample. Okay. Bring it back here, let the oil come up to the surface, photograph it, call the Coast Guard. The Coast Guard will respond to oil slicks. I saw a slick along the wall as you paddle north out of Honan now. It was scummy. I called the Coast Guard and we can send an airplane down to take a look at it. I said, no, you won't see it from the air. It's only a couple feet wide and about 2 miles long, but it's scummy. I said, what is this scum? You know, the cruise ships are able to discharge their waste 3 miles offshore, which is nothing.
There's no pumping station here.
No.
They don't go down to the pier to pump it.
If they're smart, they're going to wait till they're really in the open ocean. But out here, there's something you've probably heard of called the Kona eddy. There's a big air circulation out here. That's one reason why we're foggy most of the time, is this Kona eddy. And the eddy will bring surface water back towards shore. Prevailing wind is right out there, isn't it? Almost every day. You could count on it. And so water that's offshore is going to get blown onshore. And sewage is fresh water, and it floats on seawater. I've taken samples of this scum, and I couldn't get anything to grow. But my ability to grow everything is nil. I can grow a few things. But it sits on the surface, and so the sunlight has really got it. So you're probably not going to find bacteria. But oil, that's not going to break down. And so if you can identify an oil slick, that means somebody discharged something out there. And if it's big enough to photograph it. Yeah, so a waterproof camera and some sample vials, like your emergency response kit for your canoes.
Well, Rick, for For a regular old person like we're talking going out there, is there an agency responsible for water quality that you can submit the report to and expect action?
No.
Like the Department of Health. Department of Health, twice a year. That's all they do.
Yeah.
You know.
I raised concern about this ammonia at Keauhou. I called the state office. They sent young Neil Mukai out there. The people from Keauhou said, "Oh, let me tell you about that. Come over here." They completely diverted his attention from what I was saying they should pay attention to. And so we've got a problem with those in power not wanting to share the power. And it's just a matter of knowing more than they know and consistently showing them that, they start to listen. You know, I've been doing this for 19 years here. Now when I call the health department, they go, "Oh, Dr. Bennett, what can we do for you?" And sometimes they accommodate me, and sometimes they just send me off on goose chases and nothing ever happens. But they know that I've got an audience here. And now that I'm on the commission, I've got some status. So when I write letters as a commissioner, they say, "Oh, we better pay attention to this." And so your concerns can come to me, and then I can relay those on through the channels. The commission voted yesterday at the meeting, you'll read about this in today's paper, we're going to send a letter to Virginia Pressler, not to the mayor, not to the council, but to the director of public health. This is the first time that this commission's ever done that. In fact, Corporation Council said, "You know, you're supposed to report to the mayor." I said, "He'll get a copy." But we're going to make ourselves known to the director. Is there any reason we can't do that? And she said, "No, but it's just not typically done." I said, "Great, we're going to do it." Yeah. And so we're